So welcome to this presentation of AGBT and Mendelspod Live, the future of genome analysis. Welcome to the AGBT audience and to our Mendelspod audience, who will be watching this later in the week. I'm Theral Timpson. When Richard Dawkins wrote his book, The Selfish Gene, back in the 1970s, biology was focused pretty much on that, finding single genes. That was the era of just the gene, and Dawkins popularized that era of biology. But in the early 2000s, with the sequencing of the genome, we entered the age of genomics. It was about a bigger picture than that, and we discovered that genes across the genome were interacting with each other, sometimes hundreds, sometimes thousands. Biology was getting more complicated. Today, biologists want even a bigger story. We're in the age of big biology, the age of contextual biology. Some say that big biology will be as big a story as sequencing has been, which makes me very pleased to be interviewing two leaders of Bionano Genomics, the genome mapping company. Erik Holmlin, CEO, and Alka Chaubey, CMO. Welcome. So I've interviewed both of you before, I think, a couple times each, and now here you are together at Bionano. It's really great to be with both of you.
Thank you, Theral. Great to be with you.
So our slot here on the program, I noticed, was titled Virtual Happy Hour. So I guess we're the end of the day party here.
Yeah. We should stay hydrated. Stay hydrated.
It's up to us to keep it fun. So Erik, Bionano is coming off a fundraise of nearly $400 million early this year. Wow. Can I say that again? $400 million. Perhaps that's really what's meant by big biology.
There's some truth to that. There's some truth to that. Innovation is not cheap, and so capital is required. But we're at a time where people who are putting capital to work are recognizing the potential that lies ahead in genomics, and so they're getting behind companies like Bionano and many others to really give us the fuel we need to drive the big biology.
Yeah. So you can give instruments away for free now, right? Like sequencing companies.
That's not how we rehearsed it, but sure, we have money, and if people make a commitment to using reagents, we'll happily put an instrument in their lab.
But seriously, the stock's been going up. Things look like they've been very good. And it sounds like a verification of big biology and structural variation. You and I talked about it a few years ago. Why does structural variation still feel like a new thing?
I mean, it's a great question, and I have my views, and I would love to hear what Alka says. I think structural variation is a little bit of a sequencing term. I think in sequencing, we think about sequence variance, single nucleotide variance. And so naturally, when we start to think about chromosomal rearrangements, the idea of structure and structural variation comes in. In the early days of the Human Genome Project, which is why we like to remind people to go back to the map, it was a fundamental principle to think about how the genes and other functional elements of the genome were organized on the chromosomes, across some chromosomes when the genome was being put together. And I think the idea of structure and structural variation grew out of that original thinking. But cytogeneticists like Alka and other cancer biologists, people doing research and work and medical treatment in genetic diseases, hematologic malignancies, solid tumors, have been using the tools of cytogenetics for years and years and years to look for these chromosomal rearrangements. So it's not something new. The reason it feels new is that for the first time, and I think Bionano has been at the forefront of making that happen, but tools have been evolving to make it much more accessible to analyze genome structure. And so this is why we think cytogenomics and cytogenetics is reaching into its utopia phase here, because they can become a part of big biology. When it was just karyotyping, fish, microarrays, gel electrophoresis to analyze genome structure. Nobody wanted to use those in big clinical trials, but now that the Sapphire system makes that accessible the way it does, the way that other systems are approaching genome structure, now the structure analysis of the genome can be just as important as the sequence analysis.
Alka, the last time I talked to you, you were doing testing in the lab. Isn't that what you're supposed to be doing?
Absolutely. This is what I'm supposed to be doing, and this is what I will be doing.
Okay. So yeah. So tell us about why you came to work at the instrument company and what you are doing.
So when we spoke last time, Theral, I was the director of the cytogenetics laboratory at Greenwood Genetics Center. Mm-hmm. And so everything that Erik has just spoken about, one thing is that structural variations are not new, because structural variations have been analyzed or have been interrogated as a part of the genome since the 1970s. However, the issue was that there is a limitation with every technology that was adopted in the clinic for diagnosis and detection. So structural variations have been around for a very long time. So let me just, add that, in clinical diagnostics. But from there, between then and now, I also, was at another company as the head of cytogenomics, where the focus was to try to use sequencing technologies to interrogate all classes of variations that happen in the genome. So whether it is structural variations or whether it is sequencing variations. So we realized pretty quickly that there are a number of disorders and a huge part of the genome that cannot be accurately interrogated by short-read sequencing. So as part of that, people still need to diagnose and to find the genetic causes of a number of genetic disorders, whether it is cancer, whether it is children with disabilities. And this is where optical genome mapping comes in. So if you remember last time, as from the other side, as a laboratory director, I had been waiting, like my colleagues and peers, for a technology that can be the next generation cytogenomic tool that has higher resolution, that can provide more answers, and can be adopted in the clinic by laboratories that usually adopt newer technologies. So this was the impetus for me joining Bionano to drive this in the form that it can become a valid clinical tool just because of the power and potential that optical genome mapping has.
Okay. So Erik, and by the way, please either of you jump in here.
Yeah, well, I was gonna say, Theral, that there's another answer to what Alka just said, which is that she grew frustrated by our attempts to do what she described, so she decided to come and push us and really make it happen. And
okay,
there's no question that she's doing that.
So talk about that frustration and what she wanted to push for.
You're putting me in this situation, okay? So,
well, I'm curious to hear what you want to say, and I'm curious to hear what Erik wants to say about that.
So again, coming from the laboratory side, if you look at majority of the tests that that are used in clinical diagnostics, right, by certified directors, by laboratories to offer to the geneticist or physicians or pathologists for no matter which genetic disorder you choose, there are so many things that we need to put into place, regarding the specimen type, what kind of assay structure should it be, what should be the throughput that a laboratory would want, the ease of adoption, the reproducibility, the fidelity of the assay, and the ability to interpret those variations coming out of any technology appropriately. So Bionano, as you just started this conversation with the funding and everything, had been moving obviously, but now we have the leverage to really add a lot of fuel to these efforts to make progress in so many different domains that we can cater to what the customers would want in terms of adoption. So I think this is what Erik was referring to.
Okay. So yeah, so Erik, talk about some of those domains.
Sure. I mean, I think that when we think about a platform like the Sapphire system for genome analysis, and remember, this is what we're doing here is optical genome mapping, which is also an idea. David Schwartz was really the one who was coining that name, and you know, there have been other companies that have gone down this road, and our technology, which is really at its core nano channel arrays, enables the massively parallel linearization of DNA molecules. And then we use single molecule imaging technology. The optics industry is just improving every year and making it faster and faster and faster, easier and easier and easier to see these images. And so now we have these arrays of massively parallelized, linearized molecules. We labeled them, they have patterns on them like barcodes from the genome. We take a picture and we know where every one of those fits. And you know, so this technology has incredible utility. The question is, where is it going to be adopted first? And what we're actually seeing now that we have finally reached I think a critical mass in like weekly or yearly throughput. So around 96 samples per week, upwards of 5,000 samples, you know, human genomes per year can be analyzed on the Sapphire system. This is sufficient to accommodate many large research studies, translational research studies, but laboratories can adopt the system, research use only, develop an assay and validate it as a laboratory developed test, and then use it in patient testing. The question is, how would they use it? So they would be following medical guidelines, right? Today, they're following medical guidelines and using microarrays as frontline in genetic testing or karyotyping in cancer. And so the Sapphire system can actually be used as an alternative to those modalities. So it can do everything karyotyping can do and more, everything that chromosomal microarrays can do and more, and fish as well, with the exception of some rapid fish assays. So there are some fish assays that are very fast. So this becomes a compelling capability that labs can adopt. Where are they going to adopt it? Guess what? In Europe, they're adopting it very, very broadly with a focus on replacing these traditional cytogenetic assays. And so we observed this, we saw this, and we wanted to follow the same path here in the US. But what we learned is that a lab that's running a workflow and getting reimbursed for it is not always super enthusiastic about changing that workflow when the reimbursement is somewhat ambiguous. And so this is an area where we are uniquely investing, and Alka is leading an effort around a number of clinical trials to develop the data sets in 4 areas: prenatal, postnatal, constitutional genetic disorders, hematologic malignancies, so leukemias and lymphomas, and then solid tumors. And so within those 4 areas, we're building an incredible data set of analytical validity. The clinical validity and clinical utility of these biomarkers are already established. We don't need to prove that. They're being tested for every single day.
Right.
But we need to prove that the system works like those other capabilities do, illustrate that benefit to payers, and we expect to see reimbursement increase here in the US. And so then the US can start to catch up with Europe and other single payer healthcare system countries for the adoption of this technology. And what we believe actually, and we've seen so much adoption and use in research, different discovery programs over the years. But what we really believe is actually once the technology is broadly available as an LDT, for example, in labs that have developed the assay and offer it, the research use will pick up, because then if you discover something using Sapphire, there will be diagnostic testing that can
it will lead
employed.
to the
and so those things go together, and that's where the big biology comes from.
Okay, so Alka, have there been LDTs already created?
Absolutely. And prior to my joining Bionano, I actually did validate optical genome mapping for a very unique muscular dystrophy disorder that is very difficult to diagnose, and a number of very cumbersome technologies are needed, just because that part of the genome is refractory to the conventional methods and even to sequencing methods. So this is where, as Erik mentioned, that these long linear molecules come into play, and they have such a huge utility to pick up these large structural variations. So I'll tell you like a little bit of a personal thing, that almost 5-6 years ago, when I first saw optical genome mapping and these DNA molecules shooting through the nanochannels, it was one of the most beautiful technologies that I had seen. And this was, at the time when I was still at Greenwood Genetics Center. But this is something that we needed to develop and bring it to this point that these clinical applications can be adopted. Now, keep in mind that if you look at the whole continuum of care, if you start with prenatal diagnosis, right, to try to pick up aneuploidies or other microdeletions in fetuses, right now for prenatal testing, if we move on from there to postnatal, now babies are born, they may have a congenital anomaly, they may have a birth defect, they may have delays and autism, as they're growing. Then we come to leukemias and lymphomas, mostly, that happen in adult onset situation. The whole continuum of care, structural variation detection is the standard of care. And now we have this high resolution technology that can pick up so many things that fall in the limitations of these conventional methods. So there are a number of labs that have adopted and brought on for even a whole genome application. So we have a laboratory that has adopted optical genome mapping in conjunction with whole genome sequencing to have a complete genome test. And then there are other laboratories that have adopted for unique clinical applications. So you can just see that slowly we have made that headway, and now we need to make sure that we can develop more applications.
Okay. So you've talked cytogenetics here. Erik, in the opening plenary of the conference, we heard some amazing examples from Catherine Brownstein of Harvard Medical School and Boston Children. So that's an example of research and actual applications. She had an example of the neurodegenerative disease. Let's drill down some more. What are some other examples? Who are some of your biggest fans?
We have worked with different labs in a number of different areas over the years, and I think that you mentioned neurodegenerative disorders with Mark Ebert, who has been at Mayo in Jacksonville, Florida, and is on the move actually to the University of Kentucky. But he's worked in different neurodegenerative disorders, looking at very complex rearrangements that are so complex that they simply couldn't be resolved by any other method than optical genome mapping because of the structural accuracy. And by resolving those complex structures, it then drives hypotheses in the biology, such as ALS or Alzheimer's disease. And the thinking there is that the degree to which these structures and their variations start to appear more and more consistently, then you have a biomarker that you can follow and use for testing. There have been many other areas in cancer or pre-cancer, such as myelodysplastic syndrome, MD Anderson. They have a Sapphire system, and what they've been working on is looking at patients who would be coming in for a classical diagnosis, but mapping their genomes. And what they find, and this is published now, that they see all the markers that the traditional methods see, but then they see a whole host of others. Why is that important? Well, something that we know in hematologic malignancies is this idea that if you look at the Kaplan-Meier curves, it's really a tale of two cities. You have split survival. You have some patients who you have good outcomes and relatively long survival curves, and other patients who have poor outcomes, short survival curves. And yet at the very start of treatment, their molecular profiles were identical. And so by being able to pick up these additional markers, and you know, they've done this at MD Anderson, but also at Penn State, it provides a higher level of resolution or a degree of refinement as to the prognosis for that patient, and can begin to, instead of having split survival, two populations, but now give you the whole spectrum. And this is just clearly important in discovering the drivers and mechanisms of cancer and cancer biology, but then manifesting and or translating, as Erik Green would say, translating into an application for that has clinical utility. And one thing I want to mention about the technique or the technology, this idea of optical genome mapping, starting as something that looks at single molecules. So when we look at, you know, 500,000 cells, you know, and all those copies of the genome in single molecule fragments, we can look at them multiple times. And it's possible to distinguish populations within a sample, right? So this is mosaicism in genetic disorders, but we all know that in a solid tumor or even a liquid tumor, the pathogenic cell type may be present in very low abundance. And so the Saphyr system is capable of very straightforwardly getting down to as low as 1%, what we call allele fraction. And that's well below the typical limits of a clinical test. So a clinical test is 10 or 20%. But in research, you want to be able to find that one cell, you know, out of 100 that could be responsible for the phenotype that's being observed. And the Saphyr system can do this very straightforwardly, rapidly, and at low cost. So it really positions it to be a powerful tool for discovery. Those are a couple of really interesting examples that we've seen and that are being described, presented, and published.
Alka, do you want to pick up on that and talk to
Sure.
the research discoverers in the audience? Talk about it as a discovery tool, the limitations of sequencing, and what this can open up.
So I think when you mentioned Catherine Brownstein, so I'll just take it on from where Erik ended. One of the things that she's also part of is the COVID-19 Host Genome Structural Variant Consortium. And that consortium is led by Dr. Ravi Kolhe from Augusta University, and it is an international consortium. We have 70, 80 collaborators there. And using the Saphyr technology, what the main initiative is, because we are in the COVID-19 pandemic, so this definitely requires a little bit of a mention, is to look at the human genome and see what these structural variants are, because we know that the diversity in humans is also largely due to the structural variations in the human genome. So what could be at play and what could be the predisposing factors, genetic factors, that allow people to have different effects when exposed to the SARS-CoV-2 virus? Why do some of them end up in the ICU and die? And why some of them end up in the ICU and recover? And why some of them get infected but still remain asymptomatic? So that is the premise. And she touched a little bit upon how they are trying to collaborate and also looking at children that develop some multi-inflammatory systemic features after SARS-CoV-2 infection. So there's a collaborative effort going on to try and resolve the unique variants versus the common variants, and to tease out this pathway, because there are so many other human genome initiatives that have started in 2020, right after this COVID-19 pandemic. So when you look at that, the amount of literature and information from last year alone on COVID-19 is absolutely phenomenal, right? It has just been exponential. But if you look at what are the variants that have been actually identified, what is the yield of testing hundreds and thousands of individuals, and also sequencing the SARS-CoV-2 genome along with the host genome, if you look at the sequencing yield or the sequencing output, in literature, it's really not a lot. That means there is a lot more that is hidden in the human genome that people have not interrogated. So this is just one example of that initiative. The other thing that I kind of like, just to add on to what Erik was saying is I'm just going to cite one example in leukemias and lymphomas or hematological malignancies. One, there are two ways that the tumors start progressing when you look at like one class of structural variation. So either there will be a fusion protein, which is oncogenic in nature, which, one example is the BCR-ABL fusion, right? It's a fusion protein, oncogenic, and we know we have the wonder drug Gleevec for it. But there are some fusions that happen because of a translocation that bring in a proto-oncogene in the vicinity of a constitutively expressed gene. So one of those, for example, is the IGH translocations, which are very common. It's the immunoglobulin heavy gene, very well reported in all types of leukemias and lymphomas. And there are almost 40 translocation partners or more than that that are reported. So it's very difficult for a laboratory that is working on hematological malignancies to try and tease out what is that translocation partner. Now, sequencing, just because of the polymorphism and the cluster and the number of these immunoglobulin genes, cannot pick up any of these translocations or any of these SVs. So this is where optical genome mapping, just because of the long molecules and because of the labels over there and the accuracy to be able to show those translocation partners. And all of this now is being published by multiple people, both in US and Europe and rest of the world. And this kind of like showcases the utility of optical genome mapping in certain classes and certain unique applications.
Yeah, okay. Do you want to follow up on that, Erik?
Well, I'm sort of, inspired to think of one thing, and I don't think it's controversial, but in working with Alka and Ravi, with the consortium that they have put together and observing the growth, and then subsequently reading things like their NIH grant and the manuscripts that they put out, there is a belief amongst these scientists and clinicians that genome structure and variation of genome structure, genome structure and variation of genome structure is very likely to be as important. That's the sort of political way of saying it, and maybe even more important than sequence variation. And so why is that? Of course, I think that because we're the structural variation company. That's what it says there. We love structural variation. We don't focus on sequence variance, and there's a reason for that, because if you want to get to single base resolution, you have to slow the system down. So we made a conscious decision to say, well, we're not going to do single base resolution because we want to go fast. Right. It's got to be relevant for large studies, clinical use, and clinical applications of LDTs is what I mean to say. It's got to go fast. So we'll sacrifice the single base resolution. But the other thinking is that large rearrangements in the genome can impact many genes, many other functional elements in a single swath. So, like a tornado is a really big deal, and when it goes through a town, it's pretty mangled often afterwards, but a heavy windstorm doesn't often produce that effect. And so structural variation, the hypothesis is that it's driving a lot of biology. The technical challenge is that because of the repetitiveness of the human genome. It's not just the human genome. Plant genomes are even more repetitive than the human genome. The human genome is the one that gets all the funding. Good, but it's so repetitive that resolving the repetition is required to see its structure. And so, you know, I believe that the work that they're doing in this consortium around COVID, and you know how patients respond to the disease and progress, you know, these other areas of you know, we talked about neurodegenerative disorders, cancer, the amount of biology that's laying there that's been invisible. It used to be called the dark matter.
Right.
It's huge. It's big. It's substantially, you know, substantially large. Maybe larger. That's not for me to say. That's for us to figure out.
Well, I appreciate you going into that. I mean, if somebody's going to know, it's, I mean, you should have a kind of sense of that, right? With your customer base. And I understand that, you know, it's kind of an intuitive sense here. You know, you use the word, I believe, because we're just nibbling at the edges, maybe. Do you have a sense of where we are in big biology? I mean, are we just at the beginning? Or are we kind of in the middle? Are you getting a sense of that?
Yeah, I mean, you know, we have thought about it in the following way, and said that is, you know, when we looked at the first 2 decades of this century, I think clearly the 2000s were the microarray, gene expression, you know, SNPs, and next generation sequencing came in, revolutionized Sanger sequencing, and brought genome analysis, especially DNA analysis, to an incredible level, industrialized it, right? Really made it something that could go affordably and fast, so you could get populations done. And to a certain degree, that has leveled off. If we look at the outcomes, though, they're extraordinary. So, you know, I was in molecular diagnostics right out of school, and I observed Randy Scott and Genomic Health. And when we think about what Genomic Health did to create molecular diagnostics, deal with the reimbursement and all of those things, incredible, but enabled by microarrays and other genomics technologies.
But they
spawned
were
an incredible industry.
They had to be the first and the first and the first and the first. Yeah,
the first is tough, but you know, amazing. And now when we look across that industry, we see a diaspora of companies that grew out of that idea. And you know, for those people who pay attention to the stock market, are very handsomely valued right now. So that whole industry came out of those first 2 decades. But look at adaptive biotechnologies and sequencing all of the T-cell receptors. This is something that you know, NGS and genomics has driven. So yeah, but I do believe it's fair to say that now the focus is much more on content and applications of the technology. Those technologies are not as advancing at as fast a rate. Illumina has acquired Grail. They have an intense focus on content and application. So big biology is going to come somewhere else. We know about spatial biology. It's incredible. Single cell biology. But within the genome, within the nucleus of the cell, structural variation is it. So we feel we're very much at the beginning of the movie. We think it's going to be an incredible movie. And we're excited to be at the forefront in helping it unfold. But we're very much at the beginning. And that creates a unique challenge for scientists who want to work in this space, because when you do sequencing and you know, you do resequencing and you find single nucleotide variants, there is an abundance of databases out there for which you can look for that variant and see if somebody else has seen it and describe it in the literature. There has been no tool that allows that systematic discovery and cataloging of structural variations. One of the most important steps in that process was taken last week by you know, Charles Lee, Jan Corbell, Evan Eichler, who are the co-chairs of the 1,000 Genomes Consortium, which has been focusing on structural variation for over a decade. And they published a paper,
character in this movie.
big players, and they published an incredible paper Bionano was part of it. Long read sequencing was a part of it. Illumina sequencing was a part of it. And the most complete collection of genome variants ever, spanning ethnicities. And this is the starting point. There's some 100,000 or so variants in there. And so this is really the start of having the information to begin to rationalize that N+1 discovery. And so when we build that infrastructure for discovery, which is ongoing, those guys have done it, but there are other consortia working at this. Ravi's consortia is looking in a narrow field, but pretty topical: COVID susceptibility and COVID response, host genome response. We'll build these databases, and that will enable this wave to build and build and build. And it seems like an incredible opportunity for discovery, and it seems like it has to be the area of big biology. And one thing that's unique about it, when you compare it back to the first 20 years of the century,
okay,
is that this, the era of structural variation is coming off a foundation of structural variation driving so much clinical practice already today. SNPs were not such a big deal. We're not such a big part of mainstream medicine, but today, if you have a genetic disease, you're getting chromosome microarrays. If you have cancer, you're going to be karyotyped. And so now these technologies will build on that foundation. So
that's true. There are some tracks that have already been laid, right? There are a lot of tracks.
Absolutely.
Yeah.
Absolutely.
So Alka, we're over halfway. You're very passionate on this topic. This is AGBT. Have you said what you want to say? Erik said the movie has yet to play out. How do you see the movie?
I completely agree with Erik. And one of the reasons is, as I mentioned, karyotyping and has been in use since when? Like 1970s. The reason why it's been there, right? And then FISH evolved and standards and guidelines came in. Remember, there is no FDA approved or cleared for karyotyping or FISH. I mean, those assays are all LDTs that have been used since 1980s and 90s, right? For the most part, there are a few FISH ones for some applications, but the majority of them. And then came microarrays in 2010 that were adopted by medical associations such as American College of Medical Genetics, American Academy of Pediatrics, American Academy of Neurology. And those set the pace for what should geneticists, pediatricians, neurologists, pathologists, what should they do when they are faced in a situation when a patient shows up with specific clinical indications, and what testing should they follow for patient management and care. So this is what I have learned all along, and this is what I have been part of. And I've seen the evolution of, Sanger sequencing being used for single gene disorders or sequencing variants, and then moving on to next generation sequencing, again, around 2010 and so, and then exomes and now genomes. And every time a technology came in, it was like, well, we're going to take care of structural variations too, and these technologies may not exist or will be obliterated. But guess what? Cytogeneticists have evolved into cytogenomicists, which is what we call as a next generation cytogenomic revolution. However, we realize that all of these emerging technologies that are becoming the standard of care, if we look at even prenatal diagnostics, right? So the International Society of Ultrasound and Obstetrics and Gynecology or American College of Obstetrics and Gynecology, all of them have recommendations and guidelines for confirmatory diagnostic testing. And all of them cater to the detection of structural variations. So now if we look from 1970 to 2021, where we are, what is this era and what are we going to see now? We are going to see all of those hidden parts of the genome. We are going to be able to identify in terms of discovery and diagnosis. And I also believe that this is just the beginning of how structural variations and optical genome mapping can be used as a tool for patient care and management. that is what we are here to do, right? For genetic testing, for betterment in terms of diagnosis, so that physicians, pathologists, oncologists can better manage their patients. So this is the reason why I've joined Bionano Genomics.
Okay, so what's the path to actually get a test reimbursed? Like, how far away from that are you guys, and what do you have to do to get there?
Well, some of the laboratory developed tests that are running on Saphyr today are reimbursed today because they're looking for analytes for which there are existing CPT codes that are analyte specific. Other CPT codes are more technology specific, and so they apply to a specific legacy or incumbent technology, and maybe not so obviously apply to the Saphyr system. And so for those types of LDTs, and a good example is just genome-wide structural variation for a genetic disorder analysis. So the comparison would be chromosomal microarray. That process involves getting the data, doing the analytical validation, and we have some of that. There are a handful of papers have been published, and we're going to drive more and more of those papers. And so getting the analytical validation, I mean, people appreciate it when I make claims about the capabilities of our technology, but they appreciate it more when we prove it in a publication. So we need to do that, and it's not just one paper, it's multiple papers, and it's also papers here that are coming from US-based institutions, because there are idiosyncrasies associated with those genomes and those samples and those programs. So a data set, and we're working on that. But it's not just a data set, it's really working with other labs or having other labs see the need for and utility for the capability, setting it up, developing LDTs, applying for their own reimbursement codes, proprietary laboratory analysis codes, and then testing patients and sending the bills to insurance companies. Because when that happens, it's analogous to what happens in the research side of things. The first person who sends a grant to the NIH hears that nobody's at the NIH has ever heard of that. It can't be any good. But when they're, which is why I left academics, but I'm sorry, God bless all these AGBT folks who are
Erik was an
fighting it out. I was, so if there is a critical mass of adoption in the field, then it starts to make sense for payers to pay more universally. Right. And so that's the path that we're on. Right. And that the market is on.
Yeah, you mentioned Genomic Health. I mean, they were a lab who got reimbursement for their tests, but of course they weren't an instrument manufacturer. They were using other instruments, right? So it'll be a lab like Greenwood Genetics Center, which will get using your instrument, which will get the test reimbursed, right?
Right.
Yeah. So, and the distinction, I would say, I mean, the distinction is pretty important, right? Because Genomic Health, looking at certain patients within breast cancer stage 2, looking at a set of biomarkers, which based on a panel that they have curated, or they curated, and looking at patient outcomes and working very hard to determine what is the right way of applying that information, adjuvant chemotherapy or not, and how does that improve outcomes in the long run. So establishing this idea of clinical validity and clinical utility. We don't contend with that, because the tests that our end users run on the Saphyr when they develop an LDT are the tests that they are running on karyotyping, right? So the clinical validity and utility is already established, and that is not something that needs to be argued about. What needs to be established is that the performance is comparable to those existing methods or better, and that there's a compelling reason to switch. And so that's what we focus our efforts on.
So Alka, you mentioned that you joined the company to kind of push them along for certain things. Do you want to talk about that, like what you'd like to see happen in the next year or two, and also what you're hearing from labs like you used to work in, and even around the world.
Absolutely. So as I mentioned, right, this optical genome mapping is a very unique technology, right? That it fills the void in so many places. Just one technology that can even enter the prenatal clinical application space to postnatal, to leukemias, lymphomas, and solid tumors. If you just look at, as I was mentioning earlier, the continuum of care, it just is an ideal technology. But there's more work to be done, absolutely, because every lab that wants a platform wants it to be cost effective, wants it to be easy, ease of adoption, wants to be able to do the analysis and interpretation in a manner that that is understandable, right? A lot of the genome or genomics or information that comes out, a lot of algorithms need to put into place before you can make sense of all of that information. What we are trying to do here is to, provide these variants in a manner that the labs can understand it, relate to it, and can immediately understand the clinical interpretation or application of it. Now, in terms of what we are trying to do here, what I would like to see in the next, in this year and the next year is exactly what Erik was alluding to. We are going to be promoting and doing a number of clinical studies with a number of our partners and early adopters of our technology so that they can develop all of these applications. And now we will be directly working with them so that we hear their voice. What do they want in their lab so that we can help them do their job better? And no matter which way we look at it, do we need to develop more sample type applications? Do we need to, how much more throughput do we need to increase? Do we need to reduce the turnaround time? What would be the most cost effective way that they get all of this high resolution genomic information and it is affordable for them? And along with that, allow them to make sense of all of this data that they have never seen before, but they can look at, relate to, and say, I get it. I know what the structural variation means. I know I can see this translocation. I don't need 20 years of experience to sit on a microscope to look at a metaphase and say, I don't know what this is, and I've not been trained on it. But to look at optical genome mapping data on the screen, and it just tells you exactly what's going on in the genome, is something that, I think that is how we'll be able to help all of our customers. So this is what I would like to see this year. There's multiple applications by multiple laboratories, and just to, move forward in that direction.
So Erik, let's end and look forward with you. Our last question, we were talking about reimbursement, made me think the other tools company, the sequencing tools companies are becoming diagnostics companies. Actually, they are seeking FDA approvals, and Illumina, you just talked about Illumina buying Grail. Illumina's turned into a diagnostics company. I'm curious, for you to look out long-term at your future, but also short-term, how are you going to spend that $400 million? I'm sure you have customers in the audience. What can they expect over the next year as well?
Sure. So, I think that our focus on performance of optical genome mapping in the Sapphire system is really kind of everything for us, and that's been kind of proven over the years. The faster we make the system, the easier it is to use, the more affordable it is, the more people there are out there who are able to adopt it and want to adopt it. So it's continuing to focus on that. So a dimension that Alka brings is this ability to do these types of studies outside, bring third-party unbiased data into the picture. And so now we have product improvement going along with sort of product development, and product improvement going on with market development to define the applications, connect the dots for end users of where and how to use the technology. So it's really a really good alignment of the outside and the inside of what we can do. To be very specific, we want to continue to increase the throughput of the system, and so we've talked about this. You know, we achieved a level about 5,000 human genomes per year. When you do cancer or you go really deep in the genome, those numbers go down a little bit, and so we just want to keep doubling down on that. That was a 14-fold improvement from the time that we launched the Saphyr system at AGBT. Lisa, I saw you earlier coming on, so thank you for the venue, as always, to be able to showcase the capabilities. But we launched that at AGBT, 364 human genomes per year, which was something that was incredible. It's now up to 5,000, 14-fold improvement. And so we're sort of going for another 14-fold. Now that's going to take place over a period of years, but we will focus on that. And so people who are adopting the technology today will enjoy additional improvements based on software and other capabilities that we bring in. Cost is something that we're very focused on, and so we want to continue to improve our own manufacturing methods so that the cost of manufacturing the product can go down continuously. It has been, but it will continue to go down. And we sort of get it, like we're not going to keep all those savings for ourselves. We're going to pass them on to the market, because we see that when these assays get more and more affordable, there's more and more adoption. So those are things that we're focusing on. So I hope people don't hear this and wait to buy the system or buy the product because, oh, we'll wait till it gets cheaper. Don't do that. Start working now, and then when the prices go down, your volume can go up. You can do 2 times more patients and so forth in your study. So those are some really important, I think, blocking and tackling initiatives along with the studies that Alka is going for. But we raised I don't know how else to call it. I mean, it's a ton of cash. We've always been capital starved. This has been a limitation of Bionano Genomics from the time that I joined. I have been the CEO for over 10 years now. I joined in January of 2011. And I didn't have any gray hairs when I joined.
Okay, so that has scaled up
said
nicely.
to me the other day, my wife the other day says, I only have gray hairs now. So, there's the natural progression, but we view it as having another decade to work on this, and that it's our calling. It's our calling to drive this forward. And so, are we going to become more of a diagnostics company? Guess what? In August of last year, we acquired a CLIA business focused on pediatric neurodevelopmental disorders. So we are selling services. It's called Lineagen. So through that business, we sell services. We have salespeople who call on pediatric neurodevelopmental specialists and pediatric genetics specialists every single day. We have tens of samples coming in the door, hundreds of samples per month of kids who are trying, and their families who are trying to figure out what is driving their biology by looking at their genome. And the reason that we did that, and maybe this is what you were talking about, is that if you want to be good at providing solutions that help diagnosing patients, you got to know something about diagnosing patients. If you want payers to pay for a test that your customers are going to be buying from you and utilizing, you ought to be talking to payers. And so through the acquisition of Lineagen, which is an incredible team based in Salt Lake City and around the country, and the sales side, we have a whole host of contracts with payers. And so we're interacting with them multiple times per day, right? It's part of our business. We're interacting with patients. We have genetic counselors. We're explaining to families what their chromosomal microarray and whole exome sequencing results mean. So we have that. And so when I think about that in the future, what I think about is when we integrate Saphyr into that business, and now you have Saphyr surrounded by whole genome sequencing, whole exome sequencing, and all of these tools. I actually don't know exactly what that's going to produce. I just know it's going to be amazing. The capabilities will be amazing. And by being fundamentally a life sciences instrumentation company, our goal is to proliferate the technology And so we're going to figure these things out. And as you can see, we're talkative. We're going to tell everybody about it. Right? So our goal is to figure stuff out and then teach people how to leverage those tools and those capabilities. That gives us a ton of joy. That gives us a ton of like motivation. We know it's all directed at helping people. And so that's why we look at the next decade and say, you know, for us, it's the decade of Bionano, but it's the decade of structural variation analysis.
Well, it's really fun to share your passion for this. And I look forward to hearing more about that, seeing that in the future. Erik, Alka, this has been a real pleasure. Thank you.
Thank you very much.
Thank you, Theral.
And thanks to the folks at AGBT and LabRoots for making this a successful presentation. Thank you, everyone.
Thank you.